Next up is David Rawlings from the Seattle Children's Research Institute. He'll be giving a talk called Engineering Human Primary Cells for Novel Clinical Applications.
Great. Thanks very much for inviting me here, and I'll get right to it. So I wear kind of a different hat. I'm an immunologist, and I take care of patients who have genetic immune deficiency diseases and autoimmunity. And so moving cell therapies into those clinical applications has sort of been my long-term goal. And over the last 10 years, we've moved from sort of viral-based gene therapy in hematopoietic cells to trying to use gene editing to make therapeutic products. And there were sort of two major things that had to happen for this to be possible. One is the advent of designer-specific nucleases, and the other key thing is figuring out a way to introduce a donor DNA template into these primary cell populations that didn't turn on a response to DNA or an innate response that killed the cells. And so what I'm going to show you today is we've sort of solved the second hurdle, and we're taking advantage of what's happened in the nuclease engineering field. Here in Seattle, we've been very good at engineering a type of homing endonuclease, which is a designer nuclease. But in our lab, we're using all of these platforms, and a lot of the work that I'll describe is done with CRISPR. So I think these platforms are familiar to you all, but one of the sort of roadblocks in the field had been when you introduce this designer nuclease, how do you get this repair template into a primary cell and get homology directed repair to happen? And what we found about 2 years ago was that if we use adeno-associated viruses to deliver the DNA template, we could get this amazing rate of homology directed repair in primary T cells. And we now move this into mobilized CD34 hematopoietic stem cells. And as an example, we've been able to introduce HIV anti-therapeutics into the CCR5 locus in T cells. We've put CARs into the CCR5 locus, chimeric antigen receptors into the TCR alpha locus. We've rescued a disease called hyper-IgM, which is a defect in primary T cells. And we have a number of ways of sort of improving the editing efficiency in these platforms. And so with this in hand, we're really on the crux of moving this into a broad range of clinical applications. And what I'm going to tell you about today is two of those applications. One where we're going to make regulatory T cells using this approach. And the other, which is completely new, is moving this into editing primary human B cells. And hopefully I have enough time to get through this. But if I don't, you can ask me questions afterwards if I missed out part of it. So the first part is FOXP3 editing to make regulatory T cells. And I want to make the point that this is a collaboration between multiple investigators at our institute. Jane Buckner at BRI has been working with us on trying to move this technology into making regulatory T cells for type 1 diabetes, and we have funding from a variety of sources for this. So in a simplistic way, the immune system is balanced between the function of activated T cells and then turning them off using what are called a regulatory T cell population. But in autoimmune diseases, the effector T cells sort of get the best of the events, and they drive this disease process. And this is true for type 1 diabetes, MS, and many other autoimmune diseases. So in the last 10 years or so, using natural Tregs delivered into patients has begun to happen. And the idea there is to try to restore tolerance by delivering a patient's own Tregs back into his body as an adoptive therapy. However, this approach has a number of limitations. First, Tregs from patients with autoimmune diseases are probably not normal to begin with. They're also relatively hard to get in large numbers and to transfer. And when they go into an inflammatory environment like this, they can exhibit instability and not function normally. And in all of these approaches are not enriched for the specificity of the antigen receptor that's required to turn off a tissue-specific disease. So we kind of took a simplistic approach to this, and we said, well, if the effectors have the specificity that we want, why don't we just turn the effectors into regulatory T cells using gene editing? And so there's a transcription factor in regulatory T cells, which is the canonical transcription factor that drives the T regulatory program. So we thought if we could turn this transcription factor on in effector T cells, we could turn them into regulatory cells. So to do that, we first took advantage of a GFP knock-in strategy. So we targeted the first exon of FOXP3, and then we made a fusion protein between FOXP3 and GFP using an AAV6 donor. And I'll point out that in normal effector T cells, this gene is turned off because the promoter activity is essentially silenced by methylation of this region called the TSDR. So we thought if we could introduce a different promoter, a heterologous promoter into this downstream of the TSDR, that maybe we could overcome this regulatory methylation. And that's exactly what happened. So when we introduce this cassette, we can see the cells become FOXP3 GFP double positive. We don't see that in mock edited cells, and we can track this molecularly. So in the interest of time, I'm not going to show you all the detailed phenotyping that's been done with these cell populations. We've used RNA-seq, and we've used a whole bunch of flow and functional assays. But essentially, these cells have the features of regulatory C cells. They have high expression of the IL-2 receptor and sensitivity to IL-2. They express these inhibitory receptors like CTLA-4 and LAG-3. They have a broad TCR repertoire because we're using a input broad repertoire of T cells. And they have very low level of inflammatory cytokine expression, but they express some of the regulatory cytokines. But one of the key things about these cells is they remain, unlike the natural Treg, which doesn't have methylation at this region, they're still fully methylated and look just like the effector T cells. So we've bypassed that regulatory process. So do they work? So this is an experiment where you take effector T cells, label them with a dye that gets diluted as they divide, and then we mix them with the edited cells or mock edited cells. As you can see, if we use the mock edited cells, these effectors continue to divide, but if we introduce the edited cells, it blocks their proliferation. So these cells express high levels of FOXP3, they look like a Treg, and they suppress in vitro. So do they function in vivo? So one of the ways that we've looked at this is using graft versus host disease model. And we know that if we take human T cells, we put them into an immune deficient mouse, they recognize the mouse as foreign and they drive this GVH process. So that's sort of shown schematically here. So the question is whether if we co-introduce these edited cells in association with these pathogenic effector cells, can we turn this process off? And here's how we do the experiment. We edit the cells, we enrich for them, and then at 3 days before we add the effector cells, we add either mock cells or edited cells, and we irradiate these mice to introduce inflammation, which promotes the GVH process. We add the effector cells, and then we follow them for up to 50 days. They begin to lose weight, and I'm not going to show you the pathology, but they get a very diffuse GVH process that's affecting both the skin and multiple tissues. So does it work? Well, these cells markedly delay this very robust GVH response, and they have increased survival, and they have much less pathology. And when we look at these animals, say at 2 weeks into the process, we see the GFP cells are getting enriched in all the tissues. They remain FOXP3 high in each of the tissues of those GFP positive cells. And then the proof is really here, where if we look at 2 weeks in the animals that get the effector only or the effector plus mock cells, we see this massive CD4 T cell expansion in the tissues. And that T cell expansion essentially abrogated, and you don't really see any T cells, the red traces here, if you look numerically in these different tissues. So these cells function in vivo, and they last up to at least to 50 days. And when we look at these GFP positive cells at day 50, they're still low for these inflammatory cytokines, and they have sustained survival in that setting. So what happens with an antigen-specific engineered regulatory T cells? So to do this, the animal models are a little more challenging, so we actually moved into a mouse multiple sclerosis model here, which is called EAE. And in this case, antigen-specific T cells, if you use this TCR, 2D2 TCR, which is specific to the myelin sheath proteins, an epitope on these proteins, if you transfer these cells and then you immunize these animals with the antigen, they get very severe paralysis within 2 weeks' time if you put them into an immune-deficient mouse. So our question was whether if we co-delivered these cells, could we, like we saw in the GVH setting, could these cells suppress the T cell priming that's happening? So in order to do this, we actually had to move back from engineering human cells to engineering mouse primary T cells, but fortunately we're able to do that. We can get shown here, this is after sorting for these cells are GFP positive. And in parallel, we made both the antigen specific and then we made a polyclonal population with using just total T cells from black 6 mice to see whether the antigen specific cells worked better than engineered polyclonal T cells. So this is the experiment. We transfer the cells, we immunize, we use pertussis to permeabilize the blood-brain barrier, and then you get look for proliferation in vivo. So here's basically the take-home message. So if we use these edited antigen-specific cells, there essentially no T cell priming is happening in the lymph node in these animals. So there's no expansion of the CD4s, the antigen-specific CD4s that we transferred. Whereas with the broader repertoire, which is not enriched for the 2D2 epitope, we see, again, less expansion, but still much more than with an antigen-specific population. And in this setting, these cells still, most importantly, remain low for the inflammatory cytokines. So these MOG-specific cells can proliferate and block T effector expansion in this EAE model. So for this part of the talk, I just want to summarize. So we've got this platform now where we can make high-level FOXP3 expressing cells from engineered effector cells. These cells have a phenotype of a regulatory cell, even though they maintain this TSDR methylation. They delay GVH using human cell model. They block EAE in an MS model. And in data which we haven't shown you in collaboration with Jane Buckner's lab, we've made antigen-specific human T regulatory cells. And these cells also function in vitro. And finally, we're now moving into ways where we can make these cells, enrich them in vitro, and then manufacture them with different technologies. And so we're now moving this forward towards application in a primary immune deficiency disease where patients lack regulatory T cells, and then subsequently into either a solid organ or a GVH model clinically. So in the second half of my talk, I want to talk about engineering plasma cells to make a protein factory. And the idea here, this work is really driven by a technician in my lab who's going to graduate school. If he comes to the MCB program, you guys should grab him. He's phenomenal, King Hung. And then this is a collaboration with my next door neighbor, Rich James, in our institute. And this work is out online now, so if I don't get through it or you get confused, you can look at it directly. So the idea about plasma cells is these are one of the longest lived immune cells in the body, right? So we know that when we get immunized with tetanus, we can have tetanus titers for up to 90 years, right? So this is a very long-lived population. They live in the bone marrow in a very specific niche. And these are protein factories, these cells. So they make about, several hundred picograms of protein per day per cell. This is vastly more than even, cell line production that's done in engineering of industrial engineered cells. And so there's, I think, a lot of potential if you could generate a plasma cell that you could engraft in the body and secrete a protein of interest. And the other cool thing about this is that we think the cell dose that's required for this is going to be relatively small. And here's sort of the data that supports that idea. So here's when a plasma cell differentiates into a long-lived plasma cell, it enters the bone marrow into a niche where it's getting specific chemokine signals and also growth signals from BAF family cytokines. And these cells persist in this niche for long periods of time. The interesting thing is that the total cell number of plasma cells is about 10 to the 9th in a human, which is a small number cells when you think about the hematopoietic cell size, the total hematopoietic compartment. And it's been estimated that something like 2 million long-lived plasma cells are what's making your long-lived tetanus antibody titers. And so we think that if you could engineer an engraft, maybe as few as 10 million or so cells might actually be therapeutically beneficial. So what could we use this for? You guys can come up with your own thoughts, but certainly delivering monoclonal antibodies, treating genetic protein deficiency diseases like hemophilia, and also we think these cell products might be useful for tolerogenic applications. So here's our vision. We isolate the B cells, we edit them, we turn them into plasma cells, we give them back, right? So can this be done? So this is the one slide where we can summarize a field in a single slide. There's 2 papers previously done with human or murine B cell editing. Both of them were really not a platform that could be used clinically. One was a Cas9 transgenic. The other was delivering Cas9 stably with a lentiviral vector. So there was no data on primary immune cells, none on HDR, and none in really a clinically relevant context. So in order to do this, we had to sort of achieve 3 things. We had to find a way to edit or activated B cells when they were cycling, a way to expand those cells, and then a culture method to turn them into plasma cells. So in the interest of time, I'm only going to really talk about number 1 here, but I can show you we can expand these cells in vitro, and they, using our differentiation methods, we can turn them into the phenotype of a long-lived plasma cell. But the key thing is, can you do anything at the beginning to get them edited? So when we first thought about this, we kind of thought about different delivery methods. And one of the things about B cells is they're very difficult to virally transduce. And we also knew that if you deliver DNA as a plasmid, it essentially kills all the cells because it turns on the innate signaling DNA sensing machinery. So we focused all of our work in on either using RNP plus AAV co-delivery or RNP plus ODN delivery. And I'll show you a little bit of this and mostly this kind of data. So RNPs, for most of you in the room probably know this already, but when we're talking about RNPs, it's essentially Cas9 protein in complex with the guide RNA. So we make a complex, we then transfect this into the B cell, and then we look at cleavage of the target. And so here's essentially we transfect on day 2 after we activate the B cells with a series of cytokines, and then we look for cleavage. So here's an example of 2 different loci, CCR5 and then BLIMP1 or PDRM1. And this is a T7 assay, and then we've also used Illumina sequencing to look at a lot more reads in these cells. And so here we have 2 guides that are hitting about 80% of the cells, leading to NETJ events in 80% of the cells. And the important thing is that these RNPs are not very toxic in the primary cells. So, and we knock down the protein as expected. So we first tested, could we deliver the RNP and then a short oligo? Here, this is about a 100 base pair oligo with a single nucleotide change we're going to track by sequencing. And as we dose up the ODNs, we see about 20% homology directed repair and about 40% NETJ indels, so a 2-to-1 ratio of NETJ to HDR in that, with that approach. So what about using this to study the biology of human B cells? So these 2 transcription factors have been shown in murine models and knockout models that they promote plasma cell development, and these 2 transcription factors function to limit plasma B cell development. But in human B cell biology, there is essentially no data as to whether these transcription factors function as they were predicted in mice. So as a quick sort of test of this idea, we wanted to see what would happen if we knocked out these proteins in our models. So we made guides to all of these. This is 5 different donors, deep sequencing. So you're seeing about 80+% disruption of each of the loci with Cas9 RNP delivery. And so if we looked at a factor that is important for driving plasma, predicted to be important for driving plasma cell development, IRF4 or BLIMP1. You can see that as we introduce these guides, we limit plasma cell development in this culture, and conversely, if we do the same experiment, we knock out PAX5 or BOC2, we see an enhancement of plasma cell development. So these guides are allowing us to look at primary human cell biology in vitro. But the key question was, could we then, now that we have good cleavage with RNPs, could we deliver a DNA template and get homology directed repair to happen in this setting? And so here we used a BLIMP1 RNP, and then we made an AAV donor. We screened AAVs and found an AAV serotype that transduces B cells at high efficiency. I'm not showing you that data. And then we activate the cells and then track HDR at a time. So we can see HDR at high efficiency in this setting, reasonably high efficiency in this setting, about 20% of the cells. And if we knock in a different protein, a different transcript where we're knocking in GFP in association with a cytokine that promotes B cell survival called BAF, either alone or with a GFP marker, we can see much higher efficiency of HDR, and that is shown here by digital PCR analysis. And when we do this, and a donor lacking homology arms, this doesn't happen. But the striking thing is here, when we do this, we get protein expression, the cells survive better, and they differentiate into plasma cells. And if I jump way ahead, and I'll just show you that we've also done this for factor 9, and we can express factor 9, but when we take these plasma cells which were edited to express BAF, and we introduce them into an immune deficient mouse, which doesn't have any of the cytokines that are normally present to allow survival of a human B cell. If we have a cell that's making its own BAF, we introduce that into this immune deficient model, those cells engraft and secrete BAF. They also secrete higher levels of antibodies than unedited cells, both IgM and IgG, and they can do this for up to at least a month in culture. So to summarize, this is I think we've shown we can get gene editing in primary B cells at high efficiency. This works better than other ways of delivering a transgene into human B cells, and we can use this approach to not only manipulate the plasma cell development, but we could potentially multiplex that with the protein that we're trying to express. And essentially, what we're trying to do now is build new therapeutic cassettes to do this with, and then go to loci that would make sense for where a B cell is using a promoter that's driving high-level protein expression in a plasma cell, and introduce our cassettes into that setting. And I'll stop there and take questions. Thank you.
So that was pretty cool. With regard to the FOXP3 reprogramming experiments, is it possible to give the engineered Tregs after the immune response is already underway and drive its suppression as opposed to preventing it in the first instance?
Yes. So we're testing that now with our engineered cells. With antigen-specific regulatory T cells, that's been shown in a number of different settings. So we think that so protecting an organ transplant, for example, that may be, if you have an antigen-specific cell, I think that you can induce tolerance even in the setting of an inflammatory response. So I think it's a bigger hurdle, but I think it's definitely, there's evidence in mouse Treg antigen-specific models that will work.
Can I get a quick second one? So, and in the second context, then, when you're using the plasma cells to produce proteins, do they continue to produce immunoglobulin?
So in these experiments, I actually went so fast at the end. They're making those engrafted cells that are secreting BAF, which is what we knocked in. They're still making IgM and IgG. I probably went really fast.
Okay, yeah.
But the concept would be we knock out IgM and IgG, and then use that locus to drive expression, or at least we're not having the competing protein expression in those cells.
Got it. Thank you.
So I think we'll probably push the protein expression up for an order of magnitude or more just by doing that. . 'Cause
Thank you, David.